In this microlecture, we're going to think carefully about DNA, focusing on the structure of nucleotides, how they are linked together to make a single strand of DNA, and how two strands are annealed with each other to form a double-stranded helical DNA molecule. Whether this material is new or is review, please use this microlecture and your reading in the textbook to prepare you for Friday's discussion of DNA replication, which will quickly move us toward an investigation of gene expression and protein synthesis over the next 3 weeks. So let's take a look at the structure of DNA. Recall that on the second day of class we said that DNA is an example of a nucleic acid, a macromolecule made up of nucleotides linked end to end in a long polymer. To begin our investigation of DNA structure, let's examine the structure of a single nucleotide. A nucleotide is comprised of 3 parts. In the middle of each nucleotide is a sugar called ribose, which contains 5 carbons and forms a ring structure. To help us talk about ribose structure, scientists have designated each of the 5 carbons in the ribose sugar with a number, 1 through 5. The carbon closest to the ester is called the 1' carbon. Going around the molecular ring, the next carbon is the 2', next 3', then 4', and then the carbon extending up from the ring is the 5' carbon. We will use this numbering system frequently when talking about DNA structure, so be certain to be familiar with it. The ribose sugars used to make nucleic acids in cells always have a hydroxyl group attached to the 3' carbon. But the nucleotides used to make DNA do not have a hydroxyl group attached to their 2' carbon. Because there's not an oxygen here, those ribose sugars lacking the 2' hydroxyl are called deoxyribose sugars. The D in DNA is because the molecule contains deoxyribose sugars. DNA equals deoxyribonucleic acid. In a few minutes, we'll compare this to the ribose sugar in another nucleic acid, RNA. The second part of a nucleotide is 1 or more phosphate groups attached to the 5' carbon of the ribose sugar. If 1 phosphate is attached, the nucleotide is a monophosphate. If 2 phosphates are attached, this is a diphosphate. If 3 are attached, it's a triphosphate. We'll come back to this nomenclature in just a minute. The third part of a nucleotide is the nitrogenous base. This is a single or double ring structure that contains a carbon and nitrogen backbone and is always attached to the 1' carbon of the deoxyribose sugar. We're not going to ask you to memorize the structures of the nitrogenous bases, but do know that there are 4 flavors of nitrogenous bases in deoxyribonucleotides. Adenine and guanine are purines with 2 rings, and cytosine and thymine are pyrimidines with a single ring. Uracil is a 3rd pyrimidine found only in RNA that we'll come back to in just a few minutes. Before we leave deoxyribonucleotide structure, let's just quickly talk about nomenclature and abbreviations for these structures. All deoxyribonucleotides start with a lowercase d to indicate that they have a deoxyribose sugar. Then the next letter is uppercase and indicates which nitrogenous base the nucleotide contains. So, for the nucleotide in this image, it is a T for thymine. Finally, the last 2 letters indicate how many phosphates are on the nucleotide. So, looking at some other structures, here we have a cytidine attached to a deoxyribose sugar and 3 phosphates. So this is DCTP for deoxycytosine triphosphate. DGMP would be deoxyguanosine monophosphate. Also note that the general abbreviation for any deoxynucleoside triphosphate is DNTP. So a DNA molecule is made by linking DNTPs together in condensation reaction after condensation reaction. Before we leave nucleotides, we'll also introduce the nucleotides in RNA molecules. These nucleotides also have a hydroxyl group on the 3' carbon of ribose, but have an additional hydroxyl on the 2' carbon. Thus, they're not lacking any oxygens and are just ribonucleotides. So the molecule shown here is UTP for uracil triphosphate. This also highlights that in RNA, the pyrimidine uracil replaces thymidine. So the 4 nitrogenous bases in RNA are adenine, cytosine, guanine, and uracil, or A, C, G, and U. What would you call this molecule? Here we have an adenine attached to a ribose sugar and 3 phosphates. That's right. This is adenosine triphosphate, ATP, the same ATP synthesized as a chemical energy source in cells. ATP is simply a ribonucleotide triphosphate. This highlights that nucleotides actually have a number of functions in cells. They can serve as carriers of genetic information when incorporated into polymers of DNA or RNA, as we'll discuss in the next unit and at the end of the semester. They can act as chemical carriers of potential energy for use by the cell, as in ATP or GTP, as you learned in the previous unit. Or they can assist in communication within and between cells, as you'll discover when we talk about cell biology in Unit 3. Understanding nucleotide and nucleic acid structure and function is essential for understanding cell function. All right, now let's take the deoxyribonucleotides we just examined and put them together to make a DNA molecule. DNA is a quite stable molecule that allows for the long-term storage of genetic information in cells. As you've likely heard before, its structure was first proposed by James Watson and Francis Crick in 1953. Not only did they propose DNA structure, which we'll talk about in just a minute, but also how this structure allows DNA to be copied each time a cell is preparing to divide, which we'll cover in class on Friday. DNA is a polymer of deoxyribonucleotides linked end to end. As these nucleotides are connected, they form what's called the sugar-phosphate backbone of the DNA molecule, formed by covalently linking the 3' hydroxyl group of one nucleotide to the 5' phosphate group on the next. There are a couple of important features to this linkage. First, the attachment of a new nucleotide always occurs at the 3' end of an existing chain of nucleotides. Second, the nucleotide that's added comes in as a deoxynucleotide triphosphate. The 3 phosphates are not depicted on the nucleotide in the image here. When the linkage is formed, 2 of the phosphates are cleaved off, releasing energy that's used to link the 3' end of one nucleotide to the 5' of the other. Third, the bond that is formed between the nucleotides is called a phosphodiester bond because it contains a phosphate attached to 2 different carbons by ester linkages, thus the term diester bond. This attachment of new nucleotides over and over to the elongating 3' end of a DNA strand generates a long single-stranded polymer. Because each new nucleotide attaches to the 3' end, we say that DNA polymerizes in a 5' to 3' direction. As you know, DNA is actually a double-stranded molecule. The two sugar phosphate backbones form parallel strands that are held together by hydrogen bonds between nitrogenous bases. Specific nitrogenous bases have the structure and chemical composition to pair with each other. Adenine and thymine associate through 2 hydrogen bonds. Guanine and cytosine form 3 hydrogen bonds. These pairs of nitrogenous bases that hydrogen bond together are said to be complementary to each other. Note that each complementary base pair has one purine, A or G, and one pyrimidine, C or T, so the 2 parallel sugar phosphate backbones maintain a constant distance between each other. Note also that the 2 strands are oriented in opposite 5' to 3' directions. In this image, one strand has its 5' end at the bottom and 3' at the top, while the other has the 5' end at the top and 3' at the bottom. We say that these paired DNA strands are antiparallel, with the 5' end of one opposite the 3' end of the other. Keep this antiparallel orientation in mind when we talk about DNA replication in our next class period. Finally, the 2 strands of DNA wrap around each other to form a double helix structure, much like a twisted ladder. This structure has about 10 base pairs per 360-degree turn. If you look carefully at the double helix, you'll see some asymmetry, resulting in a large groove or cleft wrapping around one side of the helix, and a smaller groove wrapping around the opposite side. The large groove is called the major groove, and the smaller is the minor groove, and both are important for the binding of specific proteins to the DNA that we'll talk about later in the semester. You now have spent some time thinking about nucleotide and DNA structure in preparation for our examination of DNA replication and the longer study of gene expression. If any of these concepts or structures are unfamiliar or unclear, be sure to take a careful look at your textbook and please come to talk with one of your professors with questions. See you in class.